Good evening and welcome to the 22nd Annual Whiteside Memorial Lecture, focusing on COVID-19 research and implications. I'm Neil Zarvold, Medical Director of the Whiteside Institute for Clinical Research and a physician at St. Luke's Hospital in Duluth, Minnesota. Whiteside is a collaborative partnership between the University of Minnesota Medical School in Duluth and St. Luke's Hospital Clinics and Foundation, and was founded in 1996 through a generous trust from Muriel Whiteside, with a goal of advancing clinical research in the university and St. Luke's research communities, with an emphasis in the areas of cancer, heart disease, and lung disease. In addition to providing comprehensive research services and running clinical trials with nearly 20 staff members, the Whiteside Institute has awarded nearly 75 grants totaling approximately $1 million, resulting in over 50 peer-reviewed publications, 2 patents, and nearly $10 million in leveraged grant money. Tonight, I'll be co-hosting this event with Matthew Slattery, the Research Director for the Whiteside Institute and Associate Professor at the Medical School's Duluth campus in the Department of Biomedical Sciences. So you'll be seeing both of us here alternating on the screen. Before we go further, I'd like to go over a bit of housekeeping for this Zoom webinar. For optimal viewing, we recommend viewing the webinar in full screen, and it may be helpful to close all other programs during the event. The chat function is disabled, but please feel free to submit questions using the Q&A function at the bottom of the screen at any time during the presentations. We will ask both pre-submitted as well as questions submitted during the event during the Q&A session following each presentation. Finally, this webinar will be recorded. This year's Whiteside Memorial Lecture is focusing on the COVID-19 global pandemic that is affecting nearly every aspect of our lives, including all aspects of healthcare and clinical research. We will begin with 2 10-minute presentations from Duluth-based investigators, Dr. Harmony Tyner from St. Luke's Hospital and Dr. Glenn Simmons from University of Minnesota Duluth, with a short Q&A session after each section, followed by a keynote address from our honored guest, Dr. Michael Osterholm from the University of Minnesota in Minneapolis, followed by a QA. For participants interested in CME credit for this webinar, you must complete the post-webinar evaluation at surveymonkey.com/r/Whiteside2020. This survey will open in your browser at the conclusion of the webinar and will also be included as a link in a follow-up email. And you have through this upcoming Sunday, November 22nd, to complete that survey. CME certificates will be emailed within 2 weeks. Now I'd like to introduce Dr. Harmony Tyner to give our first presentation of the evening. Dr. Tyner joined St. Luke's Hospital as an infectious diseases physician in 2016 after completing medical school at the University of Minnesota in Minneapolis and residency in internal medicine at the Mayo Clinic, followed by fellowship in infectious diseases also at the Mayo Clinic. Prior to medical school, she obtained a master's in public health degree at University of Michigan in the hospital and molecular epidemiology program. Dr. Tyner is both clinically and academically productive with multiple peer-reviewed research publications, and just over a month ago was recognized on the Duluth News Tribune's annual 20 under 40 list of people making a difference in our community. The title of her presentation is The RECOVER Study, Research on the Epidemiology of SARS-CoV-2 in Essential Response Personnel. And with that, I'll turn things over to Dr. Tyner.
Thank you so much. I'm Dr. Harmony Tyner. I'm an infectious disease physician at St. Luke's Hospital in Duluth. I'm also the principal investigator for the RECOVER study at our site here at St. Luke's Hospital. RECOVER is an acronym that stands for Research on the Epidemiology of SARS-CoV-2 and Essential Response Personnel. The objectives of this lecture are to understand the RECOVER study purpose and design, and to understand the RECOVER study serial serology sub-study purpose and design. To talk about the epidemiology of SARS-CoV-2, it's helpful to have a starting point. And this, as most of you know, began in Wuhan, China at the end of 2019. To understand how we know that, it's helpful to know a little bit about the genetics of SARS-CoV-2. SARS-CoV-2 is an RNA virus. Unlike you, me, plants, animals, and most things you think about in the living world, SARS-CoV-2 is an RNA virus, and its genetic material is made of RNA. When we replicate our genetic material, we use something called a DNA polymerase, and this has an inborn proofreading mechanism in it. When RNA replicates, it does not have any such similar proofreading mechanism. As a consequence, RNA viruses are about 1,000 to 10,000 times more prone to error than DNA organisms are. As a direct consequence of that, the virus is highly prone to genetic variation over time, which means not just over months and years, but from individual to individual infection. And at the end of 2019, when they collected virus from the first folks who were infected with this virus is essentially identical, meaning it was highly conserved and it had not passed over many generations from person to person, giving high probability to the fact that it had been introduced to humans no earlier than late 2019. So this was our starting point. After that, things moved very quickly, as you know. December 31st, there was a report of a cluster of cases of pneumonia in Wuhan, Hubei province. And by January 20th, 2020, the first case of COVID was diagnosed in the United States in Washington state. By March 1st, the first case of COVID was diagnosed in Minnesota. By March 11th, the World Health Organization had declared the outbreak a global pandemic, which means that it touched most countries in the world. By March 16th in Minnesota, bars and restaurants had closed. You knew things were getting real. By March 21st, both St. Louis County, Minnesota and Douglas County, Wisconsin reported their first cases of COVID. And by March 25th, we had a stay-at-home order issued for Minnesota. Things were moving quickly, and this was a relatively frightening time. At that point in time, we didn't have a whole lot of testing, and we were fighting what felt like a very invisible foe. And despite efforts by states and the federal government to limit transmission, the number of cases continued to grow rapidly. From February to April 2020, approximately 19% of cases reported to the CDC were among healthcare personnel. Now, this was slightly skewed because we had such limited testing. We were testing mostly those who were critically ill in the hospital and healthcare personnel who were sick. So individuals outside the hospital who were sick didn't necessarily have access to a test, so we wouldn't necessarily know if they would have been sick. So this number might be a little bit stacked, but it certainly was representative of the fact that we had a large number of healthcare personnel who were becoming sick. In addition to healthcare personnel, essential workers, including healthcare personnel, first responders, and frontline workers and essential workers were thought to have the highest risk for COVID-19. What rose in that particular setting in this land where we didn't have many tests was a major question about the epidemiology, how this infection was spreading across our population. A question that was left unanswered at the time and continues to be relatively unanswered, and we're coming up with more information every day, but how many people are infected, whether symptomatic or asymptomatic? And does everyone who swabs positive for the virus go on to develop antibodies? As you may know, an antibody is a small protein that your body develops in response to exposure to an infection. Are antibodies against SARS-CoV-2 protective against infection? In some infections, antibodies are protective. For example, if you have polio or have a polio vaccine as a child, you're protected against polio throughout the rest of your life. Unlike polio, if you have hepatitis C and you have that infection go away or are cured of it, you have antibodies against hepatitis C, but it doesn't protect you against future infections. You can go on to develop another infection with hepatitis C, even if you have antibodies against it. How long do antibodies against SARS-CoV-2 last? This question is important. Not all antibodies against infections last for forever. For polio, again, that immunity does last the majority of one's life. Unlike polio, you have to get a tetanus shot every 10 years because your vaccine, your antibodies against tetanus tend to wane over time. So we don't know how long these antibodies last. And finally, related to that last point, can people who've had COVID-19 get it again? That's a very important question. In March, the CDC issued a broad agency announcement with essentially identical objectives to those, asking to assess the incidence of laboratory-confirmed SARS-CoV-2 infections, to find out if they develop antibodies, and to find out if individuals who've been infected before can get infected again. With the help of the Whiteside Institute and with the support of St. Luke's, I submitted a study proposal to answer those questions, and we were invited to join the team that was awarded the contract as one of 6 sites in a multi-center study. This included Kaiser Permanente, University of Utah, University of Arizona, University of Miami, Baylor Scott and White, and St. Luke's Hospital. CDC contracted with Abt Associates to design a prospective longitudinal cohort study of about 3,000 essential responders across 6 sites. Prospective longitudinal cohort means you start with the population at the beginning, and over time, you follow them to see what happens. This population includes healthcare workers, first responders, and essential workers. As a part of this study, enrollees are asked to have blood tests done at the beginning, middle, and end of the study to test for antibodies against SARS-CoV-2, and to perform a self-swab about once weekly to mail that swab in to be tested for the genetic material of the virus, a PCR swab. To be enrolled in this study, individuals are asked to be adults and are asked to be working full-time, 30 hours or more, in a healthcare, first responder, or other essential occupation that can't be done from home or alone. You have to interact with other people and have direct face-to-face contact within about 3 feet with other people as part of your job responsibilities. And if you're in a study that's trying to prevent you from getting SARS-CoV-2, we ask you not to participate. To enroll our population, we started in waves. The first wave is the healthcare workers who were previously tested positive for COVID-19. We really wanted to stack the deck and include as many people who had previously had this infection as possible. We really want to know if you can get it again. In our second wave, we wanted to make sure we didn't just enroll all one type of person, so not just all 30-year-old nurses. We want to include people from all age demographics and gender demographics and different occupations. So we divided people into a younger class, 18 to 39, and then 39 and up, and then we divided them to male and female in each of those categories, and then based on occupation, resulting in 8 demographic buckets, a goal of 20 in each. I will say ladies are much more enthusiastic about participating in research. So gentlemen, if you still want to participate, we're looking for more dudes. Wave 3 essentially was open to all eligible St. Luke's healthcare workers in all clinics and hospitals, including northern Minnesota and northern Wisconsin, who are part of the St. Luke's umbrella. Enrollees were asked about demographic information, including age, gender, race, ethnicity, information about their socioeconomic status and their health history, and their self-reported health and quality of life, including medical conditions and vaccination history for influenza. We also asked for permission to do daily surveillance of their electronic medical records to make sure we caught any visits. As mentioned before, we asked them to do a blood test before, during, and after the study, and to collect Neri swabs. This could be intimidating, but I think it's important to clarify this swab isn't the one that goes all the way back and feels like it's going through the back of your head. This is a swab that we do in the mid-turbinate. So you can see in that picture, that part in the middle, right around the middle of your kind of nasal area. I like to say, even though it's a little bit crude, that it's just a little further than you're comfortable picking your own nose. And to report their symptoms on the outside of the specimen bag that they submit with this swab. And we batch ship these swabs weekly to the Marshfield Clinic for testing. And enrollees are asked to also text or they get reminders to submit these swabs. If they do develop an illness during this process, during the time that they're enrolled, including fevers, chills, cough, shortness of breath, diarrhea, all the symptoms of COVID, they're asked to provide an illness kit. Now, they were given this illness kit at the beginning, and if they do develop these symptoms, they're given 3 different tests to submit, which includes 2 types of Neri swab, one which has the media in it, one that's dry, and then one that's a saliva collection tube. You might have heard of all different kinds of these tests, but they collect all these tests together and then submit them to, again, Marshfield in Wisconsin, and are asked to report their symptoms to the study team. If the swabs do test positive, whether through the illness kits or through their weekly swabs, the team is notified. The lab actually doesn't have any personal identifiers, so all positive results are reported back to the study site, and the case is submitted from the study site to the state where the individual resides, so Wisconsin or Minnesota in our case, and the individual is asked to follow the institution's standard policy for COVID-19 infection through occupational health. They're asked to continue to submit weekly swabs. We do want to know how long the virus sheds from each individual. And about 4 weeks after they swab positive, we ask them to do a convalescent blood draw to see if they've developed antibodies. Our site is uniquely doing an additional study we're calling it the serial serology sub-study, or S4. In this, we're trying to identify the serology kinetics, meaning how quickly do antibodies develop and go away. We really would like to know what % of people develop antibodies and how long does it take to develop antibodies? How long do those antibodies last once you've developed them? And does the presence of those antibodies protect against future infection? These questions would generally be addressed by the main study. However, understanding in great detail the kinetics, the rate at which they develop and go away, very specifically could have some pretty significant implications for policy and vaccine response. As a part of this study, enrollees without a history of COVID-19 are asked to have their blood drawn for antibodies every 4 weeks, so every month. And if they previously swabbed positive, so back in March, April, May, June, July, on up, swab positive for COVID-19 or swabs positive for COVID-19 through the study, whether symptomatic or not, they're asked to transition to blood draws every 2 weeks for the remainder of the study. At present, we began enrollment in early September, and as of the time I submitted these slides last week, we had 380 out of 400. As of today, we have 399 out of 400 healthcare workers, and about 75% of those have elected to participate in the serial serology sub-study. We've also invited the Duluth Fire Department and the Duluth Transportation Authority bus drivers to participate in the study, and we're expanding that somewhat. This is such an important slide because without the participation and contribution of the Whiteside Institute for Clinical Research, this really couldn't happen. I think Muriel Whiteside would be proud of the work that that has been done in her name. I also am so thankful for St. Luke's Hospital, who's been supportive from the very first moment the study was suggested, and to my incredible team, Marilyn Odine, Jennifer Virgitz, Carly Respit, Angela Hunt, Kelly Phillips, Mary Robinson, Jessica Lundgren, Daniel Staffke, and Samantha Johnson. I am so very grateful for all of your hard work, and I'm very thankful to you all for listening to this talk tonight. Thank you.
Thanks so much, Dr. Tyner. That's really interesting. I just want to highlight to our audience that the reason that St. Luke's was chosen as one of only 6 sites in the country for this RECOVER study was because of Dr. Tyner. So we're all very glad here at St. Luke's and our Duluth community that you took the initiative with your own investigation that led to our participation in this very important national study, and we're really benefiting from this. Just for full disclosure, I myself am enrolled in the RECOVER study, so I have a personal interest in following these results. So I thank you. So we have time now for a couple of questions that have come up. So I think I'll move right into the Q&A now. One question has come in. So with the community spread of COVID basically skyrocketing in many areas of the United States, including here in Duluth, how does that impact the likelihood that healthcare workers being infected, the likelihood of healthcare workers being infected in the community versus at work? And as a part of that sub-question, will the RECOVER study investigators be able to report not just infection rates among healthcare workers, but also basically adjusted for how prevalent COVID might be in the community at the time their samples were taken?
That's an interesting question. So when we do meet weekly, we talk about how prevalent it is in our community. I don't know how to answer your question. I can tell you that we are kind of generally aware of the prevalence, and some sites have more than others in terms of infection. I think the first half of that question was, are we aware of how infection spreads in the hospital? I'm sorry.
It's just sort of how does the community prevalence impact the likelihood that healthcare workers might get infected at work versus getting infected at home, or at, their kids are in school, or they have babysitters, or they go out to a restaurant or something like that?
That's such a good question. So interestingly, I feel like things have actually flipped from what they were last spring. So back in March, April, and May, before we had universal masking in the hospital, I think we probably were at higher risk in the hospital for this infection. But we are so dedicated to PPE in the hospital. We wear masks and face shields, and this is really a dedicated thing we do. I have a feeling the community spread's actually quite a bit more of a source than it is in the hospital. We're seeing this as an impact as we go along. So to your point, it's probably really significant how high community spread is, probably even more so than what we have in the hospital. Even though there's a higher concentration of patients in the hospital, we're very careful in the hospital. You're not careful if you don't know you have it in the community.
Okay, thank you. Another question that's come in is, so the RECOVER study is looking at antibodies as a marker of immunity, but what about cell-mediated immunity?
Very good question. Currently, we're looking at antibodies. There is another sub-study that's attempting to look at the peripheral, the PBMCs, that's peripheral bone marrow, stem cells, I don't know. They're looking at cell-mediated immunity. There is a study that's doing that. I don't know as much about it. I apologize for not knowing, but I think the immunologist who's actually a part of this might have more information on that. I think it's someone who's actually a part of this right now tonight, but that is being looked at. It's something I don't have as much information about. Okay. It's a very good question though.
Okay, thanks. So Dr. Tyner, we're at the end of our time for a Q&A. I'm sorry, that's very brief, but thank you so much for speaking. And we're all looking forward to stay tuned to find out, the eventual results of the RECOVER study here and beyond. At this point, I'd like to turn things over to my co-host, Matt Slattery, our Whiteside Research Director, who will be introducing our next speaker. Thanks again, Dr. Tyner.
All right. Thank you, Niels, and thank you, Dr. Tyner. It's my pleasure now to introduce our next speaker, Dr. Glenn Simmons. Dr. Simmons received his PhD in biomedical sciences with an immunology focus from Meharry Medical College in 2013, and he completed his postdoctoral research in cancer epigenetics and metabolism at Louisiana State University Health Sciences Center and the University of Texas Southwestern Medical Center. Dr. Simmons then joined the faculty at the University of Minnesota Medical School here in Duluth as an assistant professor in the biomedical sciences department in 2017. And he's been pretty busy ever since. This year alone, and this has been a mess of a year, he has mentored 6 undergraduate researchers. He has 3 papers published or in press. And this is all reflected in the fact that he was recently awarded the University of Minnesota Clinical and Translational Science Institute's Outstanding Junior Mentor Award. Dr. Simmons is funded by multiple research grants, including a University of Minnesota Rapid Response Grant for monitoring SARS-CoV-2 levels in wastewater. And that's the work he'll be talking about today in his presentation titled Monitoring Population Level Disease Prevalence by Measuring SARS-CoV-2 in Wastewater. So with that, I will hand things off to Dr. Simmons.
Thank you, Matt, for that introduction. I'm going to try my best to not mess up the transition. The sharing function in Zoom always catches me off guard just a tad bit. Let's see. Okay. Oh, it didn't go through. That's weird. All right, let's try that again. Share and full screen. All right, there we go. All right, thank you all. If you hear my little ones in the background, I apologize. They're having fun watching a movie. So today I'm gonna give you guys a kind of a brief, like 10,000 foot view of the work we've been doing over the last several months in the Department of Biomedical Sciences here at the University of Minnesota Medical School Duluth campus. We've been monitoring SARS-CoV-2 in municipal wastewater, myself, and Dr. Richard Melvin, my collaborator on the project. And so since I don't have a bunch of time, I'm gonna try to get through and get to the juicy bits as quickly as possible. So hopefully I will give you guys a little bit of history, letting you guys know that the history of wastewater surveillance in the context of infectious diseases is pretty storied at this point. Let you all know what the scope of the work we're doing is in Minnesota, and then also kind of project out into the future where we hope things will be able to go both for COVID-19 and for future potential outbreaks. So the interesting thing about my lab is that we have an interest in health disparities and cancer biology, kind of, you know, a 1-2 punch of research interests. And so it was pretty simple for us to get really interested in studying COVID-19 from a health disparities perspective, because we were already kind of tuned in on the idea that there would be some problems with different groups within the United States as the pandemic began to take hold here in our country. And so health disparities, you know, kind of a really broad stroke overview of it. It's something that's been analyzed in some form or another in the United States for over 100 years, particularly looking at, you know, newly freed African-born, African-Americans in the United States, and looking at the different factors that come into play in terms of their overall health and well-being, and also looking at indigenous populations here in America, and understanding that over the time since, you know, it's been a formal area of study, the general health of these populations and others have, you know, generally tend to be poorer than that of the European American population here in the States. And obviously, for most people, health disparities tends to live in this space of socioeconomic concerns, but there also are biological components that we're still working very hard to uncover to this day. Now, I know most of you don't need too much of an overview of COVID-19. If you can hear that, sorry, the kids are wrestling. The numbers are just constantly growing, and to the point where we're starting to get numb, unfortunately. And I hope that that does not happen, especially for those of us who are in biomedical fields and direct patient care and the like. We need to stay vigilant in order to continue to develop ways of implementing practices, both at the bedside and also in the public health domain, to help deal with these things. And one of the things that we knew early on was that the vulnerable populations here in the United States were obviously going to probably get the brunt of this issue. And particularly when it came to engaging with the healthcare system, my group, we already knew that there were problems with just doing basic medical care. And we knew that those things would be exacerbated as the pandemic began to take hold. And so we were interested in trying to understand, are there ways for us to analyze and keep track of infection rates in these populations that have a historical distrust for the medical institutions as a whole, And we sat and we pondered and we kind of stumbled across this idea of testing wastewater. And the cool thing about that was that we were basing a lot of our idea on the idea that the SARS-CoV-1 virus, the cousin of SARS-CoV-2, actually presented with a substantial amount of time with GI issues. So patients were, basically having diarrhea, which indicated there was likely some infection in the GI tract. So that also meant that you would likely be shedding virus very early on during the course of an individual's infection or their disease, which also meant that you could track the infection in a population if that presentation was common enough. The other interesting thing was that when we looked at SARS-CoV-2, there were data out there that showed that also there were a certain number of GI manifestations that were present, but weren't really talked about a lot. And so we were just super excited about the idea of just posing this as this, kind of unbiased way of looking at the infection in the populations, especially those who are not going to have ready access to healthcare, to testing due to either economic issues or, again, the social and psychological barriers that exist between those types of patients. Now, the thing is that it's interesting and as innovative as I thought that I was being when I kind of posed this idea early in February. Just a little bit of research showed that I wasn't onto something that was completely brand new. So polio eradication actually benefited a lot from wastewater surveillance. Actually, a study that was done over a 10-year period in Israel actually showed that the strategy of looking at community wastewater was very effective for being able to maintain a certain amount of supervision over the effectiveness of vaccine strategies over that community over a substantial period of time. And in February of this year, groups in the Netherlands and at MIT both showed that you're able to detect SARS-CoV-2 pretty readily in municipal wastewater, which is something that I think was very difficult for people to wrap their mind around initially, because there are so many things that could potentially make the virus degrade, make the nucleic acid material unavailable for identification through PCR. But we've also know that there are viruses that make it completely through the process of being in the wastewater for several days. And so that was actually very promising for us to show that there was a possibility of using this strategy to, test large populations without them having to be required to come in. So we decided to hypothesize that not only would we be able to detect SARS-CoV-2 in wastewater, but we would actually be able to use those numbers that we would get through PCR amplification to then model what the infection prevalence is in a community in order to kind of be complementary and even hopefully, be a completely independent way of measuring just how successful the mitigation efforts are from a public health standpoint. So this was the thing that kind of, geared us up to diving into this. And so the project itself was kind of broken down into two component parts. Give a mango. I don't care. Thanks, son. Appreciate it. Love that. I'm sorry, I apologize for that. So the first part of the project is basically just making sure that we can get a very wide picture of what the wastewater prevalence is across the entire state of Minnesota. And initially we started with a select few sites that we were able to form these direct relationships with in order to start testing out just how well we could do this and how scalable it would be. And when we did that, we found ourselves in a pretty good position to get a very interesting snapshot of what's happening in the state that was completely independent of what was happening in terms of the clinical diagnosis. But then we eventually scaled that up. And so currently we're actually somewhere around 50 total sites across the entire state, as far north as International Falls and as far south as Worthington. The second part of our project really honed in on the idea of modeling the relationship between the number of people who are infected in a given community. This is based on the clinically verified diagnosis, and then the amount of viral particles that we're seeing in the wastewater. And in order to do this, this is the part that we felt was missing in the literature. And even to this day, only maybe 1 or 2 groups in the world have done even a reasonable job of doing this. And so we're out there right now trying to throw our hat in the ring and to show that we actually have good enough data over a long period of time that would be very good for developing statistical models. And we're working with groups down in the Twin Cities, statistical analysis groups that will help us kind of put the data together and make sure that we can get all the variables in play that will help us get models that would be useful long-term. So we test for the virus in the wastewater with the exact same protocols that are used in the clinical setting. So there's no nothing super new there. We're using the same primers, we're using similar probes, and we're using a high throughput system, which is very similar to what's happening in the diagnostic labs down in the Twin Cities right now. The things that are actually different for us is that we have to process water in order to get to the RNA material that we're going to eventually amplify, which involves basically some high-speed centrifugation and a concentration of the viral particles into a small volume, which takes us about 5 to 6 hours to do before we can actually run the PCR. At the time of, before we actually expanded our project, this is kind of what our data were looking like. So here you can see kind of a snapshot of just what the levels look like relative to one another in the state from one community to the other. So this isn't absolute levels of SARS-CoV-2, this is relative levels of CoV-2 from city to city. So you can see some cities there you may recognize, some of them you may not be familiar with. But the idea here is that depending on where you're looking, you're gonna see a very different picture based on what we're seeing through this unbiased approach from the municipal wastewater, which is actually in some cases different than what you're seeing in the clinical settings. And it's different for a very interesting reason, we suspect. So when we look at the total amount of diagnosis that are seen across the state over basically since around early March, early to mid-March, you can see that there's kind of a pattern showing up there. You know, you kind of see a peak and then it starts to drop off and then it starts to come up again. We don't currently have the latest numbers in this format. We just got them in a spreadsheet and I didn't have the ability to condense it and make it all nice and pretty. But I want you to kind of pay attention mostly to this dip right there. That's the part I want you to pay attention to, which happens around June. So when we looked at the different cities that we were analyzing, we kind of broke them down by large, medium, and small cities based on population. So a large city is going to be a city that's over 100,000, medium cities are between 10 and 100,000, and small cities are just below that. And so as you can see, the large cities seem to be pretty flat overall, but the medium and especially the small cities seem to have a bit of topography going on there. And that dip that you're seeing actually seems to track very interestingly around that June period. And if we go to the next slide here, we actually started to overlay different executive orders that were coming from the governor's office, and you actually start to see a trend. And so what we noticed was that it looks like, and we're still in the space of doing the statistics, so we can't say for sure yet, but there appears to be an association between changes that are happening in terms of, you know, kind of these big mandatory orders and what's happening in terms of the viral load that we're seeing in the wastewater. But the other thing is that when we're looking at the clinical cases and we're looking at the wastewater samples, our wastewater data is actually seeming to precede what we're seeing in the clinical cases by about a week. Now, we don't know if that's entirely due to how long it takes for the data to get uploaded onto the dashboards, but we would suspect that it might take some time before a person who may have been exposed may get a test. But a person's going to use the restroom, they're going to use the bathroom in some form or fashion, and that information gets collected without anyone asking permission. It's just out there. So what we're hoping to do at this point is to take the idea that, one, we know we can detect the virus relatively easily from wastewater. We're not using a lot of water to do this. We're taking composite samples from these different sites. We can take that and then start to track how those changes are happening and happening at different population scales. We suspect that the population density is going to be a major factor in determining how sensitive our information is going to be. But we think that based on the size of a population or the density of a population and the amount of movement in and out of those different areas, you're going to see big fluctuations. But if you're looking at a big city that's 1 million plus, you may not see as much because the amount of ebbs and flows of people may end up canceling out anything that we're seeing in terms of wastewater virus prevalence. Now, the other thing that we think is really interesting is seeing how the state policy moves, those different measures that are being made, how those have an effect on what we're seeing in the wastewater. This may be another way of indicating that the mitigation efforts that we're taking can be basically measured based on what we're seeing in the wastewater. But that's hopeful. It's a tentative thing that we're looking at. We're still very much so in the thick of things right now. So for right now, as I indicated earlier, we expanded from around 20 to 44 sites, and we're now up to about 50 sites. We're also looking at residential areas to go into our future work. We're basically breaking up the project into a phase 1 and a phase 2. So phase 1 happened between April and early August. And phase 2 actually started in September, and we're just in the middle of phase 2 right now. We're still working very diligently with the biostatisticians to help develop these models. We have a bunch of interesting ideas. I have a bunch going around in my head right now, but since I'm not a statistician, I basically have to throw those numbers and those ideas at folks who really know how to make sense of it. And at this point, it's actually very, very difficult to really figure out which things are giving you just noise and which things are giving you useful information. But hopefully, even with all of that, we're going to work on developing technology that would allow us to do similar things in terms of testing wastewater, but doing it at the site. So we don't want in the future to have to bring a sample all the way to Duluth or all the way to Minneapolis from any other part of the state or any other part of the country to test this. So we're looking at ways of basically streamlining this and making it basically into a black box of detection, so that wastewater facilities or residential facilities would be able to take samples from their own wastewater outflow, their outflow, and be able to test it and be able to determine if something is happening. And if they notice that something is positive, they may be able to make changes accordingly. And so that's kind of where we're at. Another really interesting thing that happened just this week is that we actually got some information from NIH that our big project grant is getting a little bit more of a look. So we're hoping that that means that we will get the funding, and that will carry out this project and the expansion of all these measures that I mentioned for an additional few years, and keep our lab kind of in the poop water business for a little bit longer, which is very awesome, in my opinion. So with that, I want to thank the whole team. Basically, Dr. Richard Melvin and myself, we started this. We were doing everything from the beginning to end ourselves. But with the additional funding that came in eventually, we were able to recruit several very talented individuals. So Shannon is kind of our lab manager. Dan is our extraction expert. He extracts so much RNA from wastewater, it's not funny. Ty is our shipping and handling expert. So she is kind of the UPS of the med school right now. Katie Anderson and Stephen Sundin both just joined, and they're going to help us with doing some of the work that's happening on campus. We just recently started setting up for automated sampling for some of the residential buildings and other buildings on campus to show that the technique works at that scale as well. So I'm going to thank all of the folks who've helped fund this, especially the CTSI, who allowed me to rearrange my funding structure so that I could put some money towards this before we actually got any rapid response funding. Also the Institute on Diversity and Inclusion, Equity and Inclusion, I think it's that, yeah, who were able to provide a little additional funding to help us get going when things got a little bit tight. And with that, I'll take some questions. Hopefully that I didn't go too far over time there.
All right, Dr. Simmons, not too far over time, manageably over time. We've got time for a couple of questions.
Okay.
Okay, so first of all, thank you very interesting stuff, but I'm going to, I'm just going to jump right in. There are more questions than we can actually address, so I apologize to those of you who submitted something. You might not be getting an answer tonight. But first one, you talked about your RNA extraction expert. This one's kind of related to that. The person writes, do you have a wastewater management specialist involved in your study? The reason that I ask is that I have heard that the volume of wastewater produced at any given time can vary greatly. Might this variability in wastewater flow rates impact your ability to extract and detect the presence of virus?
So the quick answer is yes, but we do have a normalization strategy so that we're accounting for the human input. There is a lower limit of detection. There's no way around that. So you may we say, for instance, we can detect virus from 30 people. Okay, if we have fewer than 30 infected people, it becomes very difficult. We have that estimate based on what we've seen in the residential setting. So usually at the city level, it's not an issue at all. And I can tell you right now, we do consult with all of the wastewater management facilities who have scientists on board. We talk about the flow rates and things like that. And we've pretty much nullified that by normalizing with another virus that's coming through the pipe at the same time. That's coming from human input.
Okay. All right, another question. Have you seen any association between virus levels in rural tourist areas and tourism season?
So there has been, so if we look at certain communities and knowing that they have a certain touristy traffic, yes, unfortunately, we have seen some of those things. So some places, their population isn't really dense initially, but as soon as the weather starts to heat up, as soon as hunting season starts to, ramp up, or, cabin season or what have you, we have seen things that look like that. We don't have the data, but we do know from the folks that represent that community, our partners, our stakeholders, we do know that those things seem to be associated.
All right, I think in the interest of time, I'm going to leave it at that. Thank you again for sharing your research with us, and we'll keep watching for updates through the medical school and the regional press. Keep it coming.
Right,
at this point, I would like to turn things back over to Niels, who will be introducing tonight's keynote speaker.
Thanks, Matt, and thanks so much, Dr. Simmons. It's now my pleasure to introduce our keynote speaker of the evening, Dr. Michael Osterholm. Dr. Osterholm is the Regents Professor, McKnight Presidential Endowed Chair in Public Health, the Director of the Center for Infectious Disease Research and Policy, Distinguished Teaching Professor in the Division of Environmental Health Sciences at the School of Public Health, a Professor in the Technological Leadership Institute at the College of Science and Engineering, and an Adjunct Professor at the Medical School, all at University of Minnesota. He's a member of the National Academy of Medicine and the Council of Foreign Relations, and was previously appointed to the National Science Advisory Board on Biosecurity and the World Economic Forum Working Group on Pandemics. He spent 24 years in the Minnesota Department of Health, including 15 years as state epidemiologist. He's the author of over 315 papers and abstracts, and is author of the New York Times bestselling 2017 book entitled Deadliest Enemy: Our War Against Killer Germs. He hosts an extremely popular podcast entitled The Osterholm Update, COVID-19, and just last week was named to President-elect Joe Biden's COVID-19 Advisory Board. We are honored to have such an acclaimed international expert joining us this evening to give Whiteside Institute's 2020 Memorial Lecture, which is entitled The COVID-19 Pandemic, The Evolving Reality. Thank you, Dr. Osterholm, and with that, I'll turn it over to you.
Thank you very much. I appreciate the opportunity to be with you tonight here as I pull up my slides. Thank you. There you go. Thank you. I appreciate that. I have to start off by saying thanks for those wonderful presentations, both Dr. Tyner and Dr. Simmons. I will actually be able to abbreviate a little bit of my talk tonight just because of the wonderful work that you've presented. The other thing I must say, it's probably a bit off topic, but this past week, as I do every year at this time, I remember Duluth with a certain heavy heart and at the same time, remarkable memory. And that is, I was in Duluth the night, staying at the Radisson Hotel, the night that the Edmund Fitzgerald went down. And every year, I can't help, after all of these years, of still remembering that night and what it was like to be in Duluth. And those who are old enough here on this, you will also remember exactly that night. It was a remarkable storm. Well, tonight, my job is to talk about COVID-19 pandemic, the evolving reality. I start out by saying I have no financial disclosures or conflicts of interest to report, with exception. And that is, as I've gotten older, I'm more vulnerable to learning. And I find that in the context here that I also probably know less about COVID-19 today than I did 6 months ago. And it seems as if every day that I learn more, it's a challenge in terms of what we all really know. So I will be very clear tonight on what we know and what we don't know based on the most current data. This is an article that I published back in Foreign Affairs in 2005 as an invited piece. I did one that was similarly in the New England Journal of Medicine about the same time, preparing for the next pandemic. And went through and really detailed more about influenza, but the whole sense of why the world was not prepared for a new, particularly viral agent that would come, respiratory transmitted agent, and what it might do. It was somewhat of note that when I was asked by Foreign Affairs this past summer to do an update and try to understand what we had done in the meantime to be better prepared for a pandemic, when I published this piece here from this summer, the paragraph you see here, time is running out to prepare for the next pandemic. We must act now with decisiveness and purpose. Someday after the next pandemic has come and gone, a commission, much like the 9/11 Commission, will be charged with determining how well government, business, and public health leaders prepared the world for the catastrophe when they had clear warning what will be the verdict. That was actually the last paragraph of my 2005 paper, now serving as the first paragraph of this one. And I just want to share that with you because if nothing else, the reality is that we were not only not prepared, but we lacked the creative imagination to consider that this might even happen. And I'll talk more about that as we go through. We cannot let that happen again. To give you some sense of what a pandemic might look like from an influenza standpoint and why the changes here are important between this and the coronavirus pandemic, is the fact that if you look at in April of 2009, the emergent H1N1 pandemic, which originated here in Mexico, we basically had 38 cases from 2 countries on April 26. Literally just 1 month later, it was in 48 countries reporting over 13,000 cases. And we know, of course, that there was much, much more activity than that. This is characteristic of a respiratory transmitted pandemic agent that basically quickly descends upon the world, and why you must be prepared in advance. I'll also comment a bit on the realism of can you control one of these agents once it starts to spread. You've already heard a lot about the coronavirus. I just want to point this out to you that we actually know a fair amount about them and that there are common causes of colds. There's at least one virus that causes a picture of a SARS-MERS-like illness. And then there, of course, is SARS, severe acute respiratory syndrome, and MERS, Middle Eastern respiratory syndrome, caused by this. And I'll come back to this from time to time because one of the things that no one had any sense of what a coronavirus pandemic might look like. This is, in fact, SARS. I had a unique opportunity to be quite involved with this investigation in the sense that at that time I had been splitting my activity between the University of Minnesota and the Department of Health and Human Services, working for that time Secretary Tommy Thompson following 9/11. And I was there in 2003 when this emerged and spent a fair amount of time. One thing that I have to say here is, and I talk about this in my book, one of the most haunting memories I have in my entire career was being on the weekly calls about this particular situation. And you'll see on the left side, it says a novel coronavirus associated with severe acute respiratory syndrome. If you look down there, you'll see in the second line, fourth name over, Carlos Urbani. Carlos was on our calls with us. He was a clinician at that time in Vietnam. And ultimately, he was on the calls over several weeks. He began to get sick, coughing, etc. And several days after his last participation, he died from SARS himself. Now, this virus was actually quickly put in the bottle, you might say, when we recognized that, in fact, transmission occurred primarily 4 to 6 days after the onset of symptoms, and we could identify the animal reservoir and, in fact, eliminate that. And once we understood that about the cases in humans, we could, of course, isolate them. The same thing was true with this new emerging pathogen in 2012 on the Arabian Peninsula, which turns out to be MERS. Unlike the SARS agent, which the primary reservoir was badger dogs and civet cats, rodents sold in the food markets of the Guangdong province that could be eliminated, this particular disease problem originates in camels. 1.7 million camels on the Arabian Peninsula, for which we'll never be putting them down as such. And so we've had to control this ongoing problem by quickly identifying human cases and hospitalizing them, isolating them. And it's been quite successful overall, just because of the mere fact that, again, people are not highly infectious in that first 4 to 5 days after onset. That has been something that early on, I think people assumed that this might very well be the case with this new emerging coronavirus. I'll say more about that in a moment. So as you've heard already, this first originated, at least in terms of recognition, in the Wuhan area the last week of December. Our group at CIDRAP, the Center for Infectious Disease Research and Policy, has a very active international news team. And we picked this up in chatter between Christmas and the 31st, actually wrote this piece about it and what was happening at the time. Then in January 3rd, we had additional information. By then, we actually had contacts in the ground at Wuhan. We were beginning to see that the number of the patients were quite ill, and that, in fact, there already seemed to be a sense that, well, there's no human-to-human spread, but we can't seem to understand what's going on. Well, that quickly changed over time. It was clear that the local Chinese government was in fact hiding a fair amount of information. We say hiding in the sense that they knew it and didn't share it. And that was not part of a national initiative. The national government themselves didn't know. By January 6th, the cluster had grown to 59 in Wuhan. And we had a test now that was being used routinely. And it was clear that this appeared to be a coronavirus, not an influenza virus. Actually, at that point, I was relieved. I thought, well, if this is just another coronavirus, we'll be able to basically mop this one up like we have the other ones. If we're influenza, all bets are off, it's gone. However, as time went on, we began to see more and more information that this was spreading. It was in Hong Kong now. We were getting a sense that it was in other areas of Asia, and that, in fact, there seemed to be a suggestion at this point that there very well may be respiratory transmission well before clinical onset. Clusters started to be confirmed. At this point, clearly evidence of major human-to-human transmission, and literally by day, things were changing rapidly. On the 17th, we were able to confirm cases that were occurring outside of China and specifically outside of Hong Kong. Thailand had cases. We also were getting information on cases in possibly 7 other different countries at this point. And it was on January 20th that we knew that the coronavirus was infecting healthcare workers. We knew that it had spread to Korea, confirmed there. And we had clear and compelling evidence at this point that transmission was occurring well before people had onset of symptoms. And the fact that healthcare workers were now getting sick. So it was actually on January 20th, putting this all together and adding it up, I actually put out an email, this is the actual email, to a group we call the CIDRAP Leadership Forum, a group of institutions, organizations that we support with critical information, everything from the World Bank to the Mayo Clinic, et cetera. And you'll note in yellow there, I said it's clear now that we will see global transmission of the virus the next week to 10 days. In short, I'm certain this will cause our next pandemic. Well, that wasn't very popular at the time. A lot of people were suggesting that this was hysterics and scaring people needlessly, but everything made sense to us that this was out of the tube. And much like we saw in 2009 with influenza, I saw no way that we could put this one back in, unlike the other coronaviruses. On the 26th of January, Helen Branswell had a great story about containing the new coronavirus may not be feasible. The momentum started to pick up, saying that, yep, this was in fact likely to be a next coronavirus. Meanwhile, WHO kept saying, we can contain this, we can contain this. I found that very difficult. It almost reminded me of somebody putting a Band-Aid on a colander and thinking you're covering all the holes. it was by far way gone. On the 29th, we now that it's more infectious than SARS. Some experts had doubts. Back then, it was going back and forth between, is this influenza-like or not? And everybody wanted to say, we got to concentrate on influenza. In fact, shortly after this, there was a full-page cartoon in a major medical journal, which kind of was directed at people like myself. And they actually laid out the challenges with coronavirus and the number of cases and deaths. And at the same time, what happens with influenza is suggesting let's get on with the real work of influenza as the important issue. We never disputed influenza was important, but people just didn't have the ability to see this, even though it was in front of their nose. Finally, the WHO declared this a public health emergency on January 30th, which in fact, we were very concerned that they weren't, because the world was still believing this could be a contained virus. I wrote an op-ed piece in the New York Times on February 24th. Is it a pandemic yet? Saying, get on with it. We can't contain this. This is long gone. We need to get into mitigation. We need to understand what we need to plan for. And I might add, one of the things I didn't mention, but probably one of the more important things I've ever done in my career, on January 20th, when I put that email out, I actually met with the leadership at the 3M company here in the Twin Cities, and shared the information I had at that time, and I told them that this was going to be the cause of the next pandemic, and that they needed to understand that. On January 21st, the morning thereof, they put every machine that they had throughout the world that makes N95 respirators on full 24/7 production. And they made millions of N95 respirators before the US government ever contacted them. And I think that, hopefully they saved people's lives in that meantime. So we were trying to get them to get on with it. By February 24th, we're up now to 72,000 cases. Wouldn't that seem like a great place to be today? The actual death rate was a debate. Was it 2%, 4%, whatever? People were citing 2.3% out of Wuhan, which ironically turns out to be potentially not a far off number. Finally, the WHO raised the global risk factor to the highest level, not a pandemic, on February 28th, again, still really promoting the idea that countries could contain it. And then finally, on March 11th, almost 2 months after we first said it was a pandemic, they declared it as such. And then from there, I think you know the rest of the story. We had major problems with testing. We didn't really know what was going on, as you've already heard from Dr. Tyner. This is in the op-ed piece in the New York Times. It said, but there's a lot we could do. We need a national plan. We need to lay out how we're going to fix the testing. We need to know what we're going to do to respond in terms of our critical stockpiles of protective equipment, how we're going to plan in hospitals, et cetera, et cetera. Unfortunately, a lot of that never happened. We actually put a viewpoint out on April 30th from our website. Note the authors here include Chris Moore from our group, but Mark Lipsitch, who is a well-known modeler and infectious disease expert at Harvard, John Barry, the author of the 1918 book on influenza, and a historian who understands pandemics probably better than anyone, and myself. And what we laid out is that this is not influenza. So what's going to happen? With influenza, there have been 10 pandemics in the last 250 years. 2 started in our winter, 3 in the spring, 2 in our summer, and 3 in our fall. Every one of them had an initial wave or a peak of cases that occurred and then dropped off precipitously through no actions of humans. We had nothing to do with it. If you look at the 2009 H1N1, that's exactly what happened. I showed you those slides of it emerging in late March into April. It peaked in late April. It came down by middle of May. It was coming way down, quiet through the summer, and then a second wave hit in late August, peaking in late September, coming down throughout the first 2 weeks of October, well before a vaccine arrived. So there's something about the biology of the virus that caused it to do that. Here we had no idea what it was going to do. And so we put out several scenarios. The first one is kind of a peaks and valley kind of thing, just up and down, meaning there's hot spots and then it kind of cools down. The second one was a fall peak. This is more like influenza, where the fall peak was meant to be displaced about 6 to 7 months after the initial first peak. And then there was the third one, kind of the slow burn. What we recognize today is that in fact, this is really a coronavirus forest fire. And that basically, as long as there's human wood out there, it will burn, and it will burn, and it will burn. And to date, our best guess is, and just we're revising these numbers in the last week, even with all the activity we've seen in the United States, we're probably at no higher than 15, at most 16% of the population having been infected to date, which really gives you a sense we have a lot of wood left to burn. Why does it come up and go down, as some people would say it has? Well, it doesn't have much to do with the quote unquote biology of the virus. It has a lot to do with the sociology of humans. When we saw that first initial peak back in April in places like New York City, the Lombardy region of Italy, there were extensive efforts taken to quote unquote flatten the curve. You know, the idea of lockdowns, things like this that, you know, the terrible term, which no one can actually tell you what it means, and to really cause distancing to occur. In the United States, that peak in April at 32,000 cases a day went to 22,000 cases a day by Memorial Day. And then pandemic fatigue started to set in, and people kind of went out about themselves of the summer. We saw a second peak as such in mid to late July, involving primarily areas like Georgia, Florida, Arizona, Texas, and California, where we got up to about 70,000 cases per day, again, kicking in more of the distancing activities, the news media covering it heavily in those areas that were most impacted. And we got it down to 32,000 cases by Labor Day. Now, mind you, 32,000 cases Labor Day was as high as it was when the house was on fire in April. We just had become used to that number. Well, as you know, today it's changed very much. And I think that, as I'll say in a minute, these case numbers, I don't know where the ceiling is right now. If you look at Europe, what happened in April, they put down the brake on their outbreaks there, and they kept the brake on through much of the summer. And we're letting up gradually and actually doing a quite good job, only to find that then they let up quickly and suddenly Europe came back. In other words, again, it wasn't the virus, it was our behavior. Now in Asia, they've done a remarkable job of not only containing it through extensive efforts of testing, contact tracing, quarantine, and isolation, but it's also been done by those autocratic governments that you can say, you know, can control the people, like China and so forth. But look at New Zealand and Australia. Surely there have been challenges in those countries. It keeps coming back, but they have controlled it remarkably well. So this thing is just going to keep burning until we either develop herd immunity through natural infection or through vaccination. Herd immunity in this case would be that concept of having enough people immune, either through one of those two ways, that basically it's like rods in the virus transmission reaction, and you can slow it down. Once you hit herd immunity of 50 to 70%, it doesn't stop transmission, it just slows it down. Oh, sorry, I don't know what happened there. Come back again. Huh. Okay. I apologize. So here we are now. Somehow we've lost slides here. Let me go back in. I need to find another set of slides that we don't have on here. There we go. Dr. Simmons, it happens to all of us. Okay, now here we are. Here's where we are today. If we look at the issue of the transmission around the world, you can see that, yes.
We aren't able to see your screen.
Okay, let me try that again. Let me get out of this. It's possible I'm not sharing my screen. Thank you. Let me minimize this. How?
There we go.
Thank you. Okay. I don't think I can back up here, but Let me back up if I can, I'm sorry. Now we should be set, thank you. I apologize for the delay. So here's where we are today in terms of graphic distribution of cases. I can't quite see it because of the bars over here. Let me move that. But you can see case numbers increasing both in North America. You can see down here where we see the case transmission and what's happening. And frankly, we're now in what we call in many areas of the world exponential growth. If you look at the cases in the United States, this is a map for data from yesterday. I mentioned this early peak, flattened a little bit, second peak. Right now, 166 new cases, 166,000 new cases yesterday, an 82% change over the 14-day period, 796 deaths. Today, we had 167,000 new cases. We had 1,700 new deaths reported, and hospitalizations are now at 77,000 hospitalizations. We are now in this very dangerous exponential growth period that is going to take a very extreme, I think, and concerted effort to knock this down. If we look to give a sense of this, here's on October 30th, 2020. You can see what the red being the most intensity of cases. Now remember, this is just 2 weeks ago, 3 weeks ago. Look at it today. That's what's happened in a little over 2.5 weeks. And this is our worst fear. I had actually written a piece in July indicating that my worst fear was that this would all come together, a combination of pandemic fatigue, people who really believe that the pandemic is real, but are just tired of dealing with it and decide that they're just going to continue, whether it's the bars and restaurants, going to theaters, going to churches, weddings and funerals, going to gymnasiums, sporting events in high school, ice hockey, one of the most dangerous of the sports, which we can talk about in the Q&A section. Why? And we're now in a period where it's just like we saw in New York City, except in much of the country. And right now, all 50 states are seeing major increase in cases. The upper Midwest happens to be well ahead of the rest of the country, but we see the rest of the country coming. Clearly, we are now in a position where we're also beginning to overwhelm our healthcare systems. I will come back to that in a minute. If we look at deaths, a delayed indicator of what's going on, you can see the big peak here before we really knew how to take care of these patients. One of the really untold stories has been the incredible success of the intensive care medicine groups who have taught us so much about how to better care for these patients, and not based on a blockbuster new drug, which is better care. You can see how the number of deaths has dropped over time, coming up somewhat here with increased cases, coming back down. But I think now we're on this very steep curve up. Not because we still don't know these lessons from over here, but two things. One is just the sheer number of cases. But in addition, we're having a very hard time staffing many of our institutions. I noted in a conversation earlier before we got on here today, 900 healthcare workers in the Mayo Clinic system, 900 have been infected in the last 8 days, 900. I just talked to the group of case at Cleveland Clinic this afternoon, the same thing is true. So between the need for expanded care and the lack of healthcare workers, particularly intensivists, particularly people that have that skill set to deal with this, we're in trouble. In terms of the survival, this is what I was talking about. This is a study that was published recently showing the improved survival based on just the improvement in overall care and how both the high dependency unit and ICU dependence case outcomes have improved dramatically. Also, we see things like the new drugs. I share this one with Regeneron from October 30th. These drugs can surely have major impact, but they're going to be challenged in how they're going to be used. We now know that they have to be given very early into the clinical course, and with 160,000 new patients a day, how do you know who to give it to? Also, our stocks of this are limited, such that 1 or 2 days worth of cases could blow through the entire stockpile and even the most aggressive production possibilities. So I don't see this drug being, for most people, any kind of an important drug. And you've heard already about disparities. This is a huge issue. I just want to point out here that this is a paper from the CDC. I want to draw your attention to the mean of estimated differences here. And you can see down here that Hispanic, Latino, Black and African American, American Indian, and Alaska Native have almost a 4-fold, up to an 8-fold for Native Hawaiian and other Pacific Islander incidence of cases over the white population. This has to do with socioeconomic status, living conditions, as much as anything that we see. Europe is clearly in trouble right now. They are in lockdowns in many of the countries. We can talk more about that if we care, but they have confronted what we're confronting, and the only way that they've brought it down basically is to virtually shut down their economies. So let me just very briefly touch on a couple of issues, just I think in terms of this. One is transmission. Clearly, there's been a lot of debate how this is transmitted, but we have no doubt now that the airborne route with aerosols is very important. The concept of the super spreading events we're seeing, and it's very important to understand that in some ways, I think we approach this as if there is a one way. And this reminds me more of your head's in the freezer, your feet are in the oven, and average your temperatures just right. We do see some settings with some individuals where we have much more transmission. But today we're finding, even in households where it's not big spreading events, still we're seeing 20 to 40% attack rates, which are really fueling a lot of this outbreak right now. So we have to understand that airborne is a very important part of this. In terms of immunity, we've already heard a bit already from Dr. Tyner about this. Durable immunity is a huge question. This is the antibodies in a large nationwide study in dialysis patients. You can see the different percentages that are out there. Places like some areas of New York, actually maybe 40 to 50%, overall state average 33%. And these data are relatively recent, just suggesting that, in fact, again, we have a lot of very vulnerable people out there yet in terms of this. There's a paper that is yet to be published, but it's summarized today in the New York Times, indicating that there may be more durable immunity than we once thought. I think this is still a question that we have to raise as time goes on, whether it be through natural infection or through vaccination. This is a paper here looking at the prevalence antibody, again, suggesting that only 4.4% of the adults had detectable IgG, when in fact originally it was 6.6. So we're getting conflicting data, just how well does this antibody and of course cellular immunity play a role down the road. And this is something we're going to be learning a lot about. Also, reinfections are there. I now know of 28 cases of reinfection, which you can say, well, that's not very many. I think it's a lot, only because to be classified as a reinfection, you have to have the isolate or the swab from your first illness so that we can compare the viral genetics from that with your second illness. And the vast majority of people do not have a swab available, or for that matter, an isolate from their first illness when they get infected a second time, on average 4 months later. And with these cases, I think it is just a reminder that we don't yet quite understand durability. Even the data we're talking about with vaccines today, the data only through 2 months following the last dose of vaccine. So this will be an important discussion in terms of what we do long term. The other thing is the long haulers issue is surely an important concern. We're now seeing that we get a number of people who report conditions where even though they had mild illness at the time of the onset of their infection and symptoms, at 4 to 6 weeks later, they actually are experiencing severe fatigue, a chronic fatigue syndrome-like picture with classic brain fog. And this is something that at least 2 studies now have indicated it may be as high as 20 to 25%. So this is going to be an important challenge. This is part of that long haulers issue. We clearly see cardiac involvement. We see pulmonary involvement. There are now at least 4 new centers that have established themselves just to look at the issue of long haulers. and what type of immunologic issues are going on here. There's no evidence of ongoing viral infection. In terms of vaccines, we've had so much happen just in the last week. This is the number of vaccines. We actually have 50 vaccines right now being tracked in 117 trials from 32 countries. Operation Warp Speed surely has a portion of those. The COVAX, the WHO-led initiative, has another group of them. But it's been remarkable what's happened in the past year to get us this. And I do think that Operation Warp Speed will one day go down as a Manhattan-like project relative to getting us new vaccines. It's still, we've got a long ways to go to get those vaccines into people's arms to make them a vaccination, but we've done a lot already to get vaccines. Pfizer, as you know, had its vaccine results announced last week. And actually, I was somewhat, how should I say it, modified in my response to this, because they talked about a 90% efficacy, which surely exceeded what many people thought. But again, remember, it was at 2 months following the last dose. But more importantly, I wanted to know, did it in fact protect against severe disease? You know, it's great to protect 90% of the people from fever, muscle aches, and chills, and some diarrhea, but did you actually also prevent severe disease? Well, this morning, those data were announced by Pfizer, and in fact, there is clear evidence that it did have significant protection against severe disease. The same thing this past week at the Moderna vaccine. And by the way, the Pfizer now is up their estimate to 95% efficacy with additional data. The Moderna vaccine, both of them mRNA vaccines, also announced their results this past week, 94.5%. And what was also very important here is that they also were able to demonstrate significant protection against severe disease. This is all really good news. These vaccines, however, will not be here for at least several months in any meaningful way. We'll start seeing some use of vaccine, I think, by early next month, possibly, particularly in the healthcare worker population. And then after that, we're talking anywhere from 20 to potentially 40 million doses a month for at least a short term for both of these two vaccines in the United States. Well, if you're giving 2 doses, you can see it's still going to take us a while to get the population vaccinated. And however they can speed up production will be a key issue. One of the challenges we have, however, is that in fact, people are skeptical of COVID vaccines. We saw a poll just come out yesterday afternoon from Gallup saying that now it's gone from about 48% to about 55% of the population said that they would get this vaccine. This is a challenge. If we don't get people vaccinated, we obviously can't protect them, and it really cuts into our concept of herd immunity. And so it's going to be important that we have a very comprehensive program set up to try to deal with this issue, for which at this point we do not have that. In terms of, let me just finish off the shortage trifecta. I've already talked about the worker issue. I fear more than I can tell you that what's going to finally help the citizens of this country understand what's going on is when they start seeing hospitals literally come to a breaking point. And some of them are there right now. I just did a talk this morning for a large group hospital out in Billings, Montana, where they are breaking right now, literally. They just don't have enough employees. The employees they have are burnt out. You know, they've been working 16 hours for so long. You know, this is going to be a challenge. People are going to start dying in emergency room, waiting rooms, waiting for a bed, waiting for care. The fact that we have at this point, you know, 160 to 180,000 cases a day, this number is going to go substantially higher, much higher at this point. The next 3 weeks are already baked into the system, meaning that the transmission that has occurred this week will manifest itself as infections next week, a short period of illness before hospitalization, then moving on to the issue of ICU care, and then the time there. So for the next 3 weeks, we're really already covered. Hospitals are reeling under this spike. We're seeing it throughout the upper Midwest. You know, we're going to start seeing more and more refrigerated trucks for morgues. And the tipping point is getting close. And you all know it better than I. I think Duluth has been somewhat spared of the severest, although Duluth has been surely negatively impacted. Your police department knows that. But I think from the standpoint of where we're going yet, the worst is yet to come. And so we're going to be talking about rationing care. That's as simple as it's going to get. PPE, second of the trifecta, is still a challenge. I can promise you right now that gloves, gowns, and N95 respirators and surgical masks are going to get in short supply. Unlike what happened in July, where we were able to meet the needs to a very real degree, the surge was much, much less. And number two, Europe was not on fire also. They're ordering, they're competing for these very same PPE issues. So expect that in the near future, we're going to run out of a lot of supplies. Also, in terms of that, this is a lost on the front line piece that The Guardian does. At this point, 1,336 healthcare workers who have died from coronavirus. We know that not nearly all of these are occupation related at all. But in fact, the bottom line is it's taken a huge hit in healthcare workers for those who are at work and also those who are exposed in the community. 2 days ago, I announced with the St. Paul and Minnesota Foundation a new fund, the Front Lives Families Fund, that we've launched, that we're working to raise millions of dollars to support the families of the 1,400 U.S. healthcare workers, frontline healthcare workers who have died. I can talk more about that later. But in many of these cases, these are not physicians who had large life insurance policies. They're physicians who may have been, more recently out of training or in early days of practice, nurses, all the other staff members in the hospital, from the care team to the janitorial support team, et cetera. We're here to try to support them. This is the book that was mentioned before. I only show this because early on, I tried to make the case that we should have been aware of this. Chapter 13 in this book is called SARS and MERS, a harbinger of things to come. This was again published in 2017. We didn't own any unique space here. We didn't own any kind of magical thinking. I don't know why the world was so surprised when we saw what we've seen here. And what we can't do is let that happen again. We have now got in this world about 23 billion chickens to feed the 8 billion people on the face of the earth. Half of all the body weight in avian species in the world today are chickens. 390 million pigs to feed the pork needs of the world, and many of those pigs are living in close proximity to the chickens. You could not have devised a more competent and unfortunately very productive cauldron for making new flu viruses. The avian species infecting pigs, as well as the human viruses infecting pigs, and looking at what happens with reassortment. So as much as we can't take our eye off this very serious problem we have right now, one that I only see getting worse for the time being, we can't forget about tomorrow. At this point, states are trying to institute new measures. While we were on this conference, Governor Walz announced at 6 o'clock a number of new efforts to try to limit transmission. I'll just say that I think that, closing bars at 10 may have some impact, closing gymnasiums. But as the Europeans have demonstrated to us, and as we saw this summer where we really had to bring down case transmission, extreme measures had to be taken. I don't know how else we're going to get through this. I think that, we are kind of nibbling at the edges. I worry that the transmission will continue largely unfettered for several more weeks to months to go. I think what will finally bring it down is when people realize that they have loved ones who have been infected and who die. They see what's happening to our healthcare system. But until then, we have a lot of people out there that are very indifferent to doing anything to change what they're doing, because they either are tired of this virus, even though the virus is not tired of them, or they just don't believe it's real. So as Lewis Carroll once said, if you don't know where you're going, any road will get you there. Right now we got 50 governors, a bunch of mayors, all trying to figure out where to go. There is no national leadership right now on this. There is none. There's no one helping to direct what can we do, what should we do, how would we do it? How do we standardize response? What does it take to actually drive transmission down? Remember, we're just trying to get to the days of vaccine. We don't have much further to go. And if we don't do something, though, in the next days, I'm afraid through the holiday season, the kinds of numbers we could be seeing nationwide are scary. We're going to look back one day and say, boy, I wish we're at 160,000 cases a day. Sir Winston Churchill once said, it's no use saying we're doing our best. You've got to succeed in doing what is necessary. Right now, I'm not sure we're even doing our best. And I know for certain we're not doing what is necessary. And I'm afraid that you all are on the tip of the spear in that regard, particularly in the healthcare side. But I'm an optimist. You know, I believe we can turn things around, or I guess I wouldn't be in this business. Are these the shadows of things will be, or are they the shadows of things that may be only? And, you know, we need national leadership right now. I'm not naive to think that it's going to suddenly emerge. That's not a political statement. That's just balls and strikes. The incoming president-elect cannot really do anything of real nature until January 20th, other than help us as an FDR-like figure to understand the depth and breadth of what's happening right now and what we might do to try to get out of it. So with that, I'm very happy to close, and thank you all very much for the opportunity to be with you here tonight.
All right, well, thank you very much for not only a straightforward and sobering breakdown of our current situation, but also for ending on an optimistic note. and also thank you for your foresight and your dedication across your career for helping the world deal with this, whether or not the world wants to listen, right? In the interest of time, I'm just going to jump right into a few questions. We're supposed to stop at 7:30. If you are able to stick around for a few extra minutes, then we will keep asking, but I won't keep you on too long. Okay, so one question that was posted as you were talking is, I truly appreciate the focus on the fact that the virus does not care, but humans may make a difference in their behavior. You covered strategies to mitigate. How can we as community members help others to see the light?
Well, the single most important thing we can do is stop swapping air. It's that simple. That's what we're doing. You know, we've had far too much hygiene theater brought to this. There's no compelling evidence at all that surfaces, the environment plays any meaningful role at all in transmission. It's really about air. And what we forget is who we share air with. We have seen so many outbreaks. In fact, you know, I worry desperately about Thanksgiving. I can't tell you how many emails I've gotten from people who are just saying, well, but we're only going to limit it to 12 people. Well, right now, given the statistical probability of someone in our communities being infected, there's a good chance that there's somebody in one of those 12 that are actually carrying the virus, whether they know it or not, and could very well infect the people at Thanksgiving. I'm waiting to see what the impact has been over the course of the last 2 weeks with Minnesota's deer hunting season. We've had 720,000 Minnesotans who are out in the field. That's a pretty safe bet doing that. But the nights in the hunting cabins, where we have had many individuals spending days doing that, I'm sure has been a super spreading event kind of phenomena. So I think that the challenge we have is helping people understand what that means. And, you know, it is tough. It's tough to be isolated. You know, I'm still on my same tank of gas since early September. You know, that's how sequestered some people like us are. Now, not everyone can do that. In fact, if you're in the healthcare setting, you're an essential worker, you can't do that. But how can we limit people? How can we, you know, keep them, if the bar is closed, do they just go to somebody's house? Do you know how many outbreaks we've had recently just in people who are getting together for Sunday football game watching? 10, 15 guys getting together without any sense. Well, I know all these people, so therefore they can't infect me. You know, the issues of how many times funerals and weddings have turned into major events. I can't tell you. I can't make this up. Just a couple weeks ago, I had someone who called me about feeling forced to go to a wedding, a family member. There were 6 people in the family. They had to travel out of town. I said, that's not a good idea. They went. 4 of the 6 came back infected. One of those people is now in the hospital. You know, it just is happening over and over again like this. We have to have people understand that masking, I recommend it for everyone, but please understand it is far from perfect. Maybe 20, 30, 40% effective. You know, the data we're working on right now would suggest at best in that range. Now that's still a big, big plus, and you should mask. But at the same time, don't count on that as being the be all end all. If you go into large crowds, if the mask is only when it's convenient, I'm eating, I'm drinking, I'm talking, the mask comes on and off. Remember, these face cloth coverings were not meant from a face fit standpoint or a filtration standpoint to be also perfect, like an N95 respirator. And so I just think that we have to help people understand, get us through this, limit your contact. Now, the area that I've been very outspoken on, and it's caused some people consternation, is if we're closing down bars and restaurants or gyms or any number of different places, or even some areas of the healthcare system to accommodate the increase in other areas, these people should be compensated. I don't know any small business owners that don't want to do the right thing, but when their business is hanging on by a thread and we're now shutting them down for weeks at a time, when you got that single mom who's a waitress, who has 2 children at home, who, if she doesn't get paid, she's may not get to keep that roof over her house or over her head or put food on the table for her kids. The fact that we have not as a nation come to grips with that, to realize that's how we're going to help people help us get this under control. And again, we're just trying to get to the vaccine. So I'm concerned that it's not just the indifference of people to change their behaviors. Right now, I can tell you the governor has been bombarded by folks that are ready to impeach him. They're so upset with him because he's closing down high school sports. We have right now over 48 outbreaks in hockey, ice hockey, outbreaks. And because, this is a piece of work that I did many years ago, published it in the Journal of American Medical Association, when we had outbreaks with Zambonis. Either the fuel mix was too rich or too lean, and we either had nitrogen dioxide or carbon monoxide produced, and the players developed major pulmonary problems. Why? Because it turns out that an ice rink indoors has this huge dome of cold air that holds everything down inside from the top of the glass down. Nothing dissipates up, even though the rest of the building is ventilated. And so if you've got one infected person skating in that dome of cold air, it just sits in there. You might as well basically put a hose from a virus out there and spray it on the ice. That's what we're seeing. And yet, you know what? The number of people that are so upset with people like me and others for saying, hockey is a real problem. And so I think that the challenge we're going to have is just, how do we get through this? None of us want this to happen. I've said over and over again, this is our COVID year. This is our COVID year. It's not going to be like last year. It's not going to be like next year. But we got to get through this year. And I worry desperately that we will put our healthcare systems on the brink of collapse if we see a doubling of the case numbers in the next few weeks. That is surely on the table.
You've answered several questions. The hockey one came up, a few other questions. So I'm going to change gears here. Well, actually, another question that's on people's minds. Really, there are a lot of numbers out there, and we talk about community spread and all of these things, but which numbers matter? That headlines tend to focus on cases and deaths. Minnesota schools are using new cases per 100,000 people per 14 days. State of New York is focusing on test positivity. In your opinion, what are the 2 or 3 most important numbers that people should be aware of for their area?
Well, I think each number has a different contribution. It's like the weather. It's temperature, it's wind speed, it's humidity. It's all the things, okay? So any one of them by themselves wouldn't give you the definitive outcome. Test positivity, if it's done in the systematic way, so only new test positives are counted, gives you early information about what's happening. And if you are, for example, adding a lot of testing, but you're getting a lot of additional cases such it outstrips the testing, it's telling you got a lot of transmission. On the other hand, if you add a lot of testing and then the test positivity goes way down, it says you probably have a pretty stable transmission out there. You're just picking up more of what was there. I think ultimately the trailing indicators though are going to be hospitalization and deaths. It's hard to change those. And I think at this point, the only thing I would say about deaths is I think proportionally you could start to see the death numbers go up, the case fatality rate increase, particularly in ICU care or what should be ICU care, just because of the lack of ability to treat. And I think that's really going to be a huge issue going down the road is fewer and fewer healthcare workers. I don't know where you're at right now, but I can tell you that an ICU charge nurse who I know well commented to me that in their unit in an area not far from here, they had one ICU nurse per patient with COVID because of the severity. As of this week, they now have one charge nurse for 4 patients, and they have 2 float nurses and 1 nursing student to support those 4 patients. That's a scale down of care. It's going to keep happening more and more and more and more. And at what point do you start to see outcomes suffer just because that's the best they can do? That's not, again, a criticism of the healthcare system. It's the reality of what we're up against. And I think that's going to change case fatality rates. So I think those are by far the best indicators. The ones for schools, let me just say, are really meant to give some sense of what's coming relative to the challenge of providing in-class education. You know, the schools basically, first of all, should be very much divided by age. We're seeing a very different epidemiology of this infection in those in grade school, elementary school, than we are in junior high and high school. We have much, much more transmission in junior high and high school. Only limited transmission evidence in grade schools. So right there, I hate it when people talk about kids because it's very different. The second thing is that there, the numbers we're using is how much is in the community that then spills over into the school. The schools become a reflection of the community. And so it's very hard to provide education in schools when the community transmission itself is so widespread, and what happens with regard to transmission in the schools and the quarantine issues that come up and people out because of quarantine.
Okay, I've got another question, a multi-part. In the past, you've been unafraid to point out the inadequacy of our flu vaccines, in part as a call for major research to develop better vaccines, which makes sense. Do you think the promising news for the SARS-CoV-2 vaccines do you think the excitement is warranted as someone who has been willing to speak the truth in the past?
Well, first of all, let me actually say something about flu vaccine. Because of that naysayer in me and the studies we did to show the problems, we've actually seen a major change in that. I actually chair a group now that involves the largest philanthropic organizations in the world, as well as the WHO. And we've been working and have had developing a roadmap for new game-changing flu vaccines. And just in the last 4 months, we've actually had 4 multiple-day Zoom meetings on this issue for a roadmap, which over 100 organizations have been involved with and over 300 experts. So we're moving. I actually think there will be a day we're going to see game-changing flu vaccines emerge. In terms of this vaccine, I am very excited by what I think the vaccine can do initially in terms of dampening down and reducing transmission in our communities and surely protecting individuals. As I said in my presentation, I think the question we still have yet is what is durable immunity? And that's not necessarily a failure of the vaccine. It's going to be a function of we may be adding booster doses down the road. Vaccine. This is a function of coronaviruses. We've known this for a long time, that coronaviruses are very poor immunogens, and that this has been a challenge developing immunity. If you look at the MERS and SARS research, that clearly is an outcome that we found with those. So, you know, I'd say at this point, at least in the short term, meaning months and months, I think that we're in really pretty good shape, but we're going to have to figure this out down the road.
And aside from a related question, aside from vaccine skepticism, which you addressed, is there anything else that you can think of that might derail the development and implementation of vaccines? Are there any major barriers?
Well, I don't think anything that will derail the development. I mean, I think this is where Operation Warp Speed was such a unique entity. Normally, we would do vaccine research by doing first step A, and then if that's successful, we go to step B, and then B to C, and C to D, and D to E. And by the time you got the whole sequential process done, it could be 5, 7, 10 years. Well, it was done with Operation Warp Speed. They were all done simultaneously. So A, B, C, D, E, F, and G, including manufacturing the vaccine. If any one of them failed upstream, then everything that had been done downstream was invalidated and you lost all your money. When you have an unlimited pot like that, you can do that. I think that surely has served us well for this pandemic. I don't believe that there has been any shortcuts taken with safety. I think we all would like to have 5 years worth of safety data on this vaccine before we use it widely. We're not going to get that. You know, we got today 1,700 deaths today. We're going to be way back over 2,000 to 3,000 deaths a day. Just to remind you that in April, for 3 weeks, COVID-19 was the #1 cause of death in the United States. And that was at 21 to 2,200 cases a day. We will soon be back at COVID being the #1 cause of death in the United States. And I can see that lasting for a long time. So we need these vaccines. We need them. We can't shortcut safety. We can't shortcut effectiveness, efficacy. But at the same time, we have to accept the fact we're not going to have all the answers. And we're going to have to move forward with them. I personally, based on my faith in the FDA review process, the VRPEC, the outside group that reviews these, I can't wait to get in line. You know, I don't want to jump ahead of anybody. I'm not the highest risk person out there, but at the same time, when my time comes, I won't be a second late to get that vaccine. And I think that's what we have to mirror in the general public so that they see the confidence that we have in it too.
I think that's very, very useful advice. That's what a lot of people want to hear is who is going to be standing in line. Okay, well, we've gone a little bit over. I know you are a very busy man, even more so over with recent announcements. So I think I'm going to stop the Q&A there. Before saying goodbye to everyone, I'd like to thank all of tonight's speakers one more time. Dr. Osterholm, thanks for all that you do. Best of luck to you and everyone else on President-elect Biden's COVID-19 Advisory Board. Thank you, Dr. Simmons and collaborators, Dr. Melvin and others for getting the wastewater and environmental monitoring projects up and running here in the Northland. And thank you to Dr. Tyner and the entire Whiteside staff for quickly ramping up your efforts with the Recover study. And I'd also like to thank Marilyn O'Dean, Kara Haig, and Lindsay Radovich for organizing the evening, Aaron Verrell, Alyssa Dindorf, Melissa Berlaga and Sarah Backstrom for operational support, and everyone else at St. Luke's and the medical school who assisted in putting this together. And then finally, thanks to the hundreds of you who attended. We had great turnout for this. You will receive a follow-up survey via email, or there will be a link right when this closes down. So please take the time to fill this out. It's especially important for those clinicians who are seeking CME credit. And then beyond that, everyone, take care of yourselves this holiday season, stay safe, and maybe, just maybe, we'll see some of you again next year in a non-virtual Whiteside Memorial Lecture. So thank you, everyone.
Thank you.